Biochemistry Module Lecture 3, Non-Enzymatic Protein Functions and Analysis. We'll go through some cellular functions and what proteins will help with these functions. Starting first, we'll go into structural proteins. These, as the name would suggest, provide structure and stability for the overall composition of cells. These are comprised of things such as the cytoskeleton, anchoring proteins, and the extracellular matrix. Because of this, these types of proteins are usually very fibrous and durable, examples of which are listed below. Motor proteins are capable of force generation, and this is done via conformation change. Because of this, they have to have some sort of catalytic ability, and generally it's ATP that powers this movement. These are involved in things such as muscle contraction with respect to myosin, vesicle movement, and cell motility, and the proteins that are involved with these processes can be anything from the proteins in flagella, cilia, or internally kinesin and dynein. Binding proteins will bind to specific substrates. They do this by binding to these substrates and regulating these concentrations at steady states. So if I have a bunch of a substrate floating around, what these binding proteins will do is bind to excess substrate to help regulate the concentration. If the concentration is too low, they'll reverse this effect and allow the substrate out. So in a sense, this is to keep a very, very level amount of a specific substrate in the system. Cell adhesion molecules, or CAMs for short, will allow cells to bind to other surfaces, examples of which include cadherins, selectins, and integrins, which are listed below. Cadherins are groups of glycoproteins, and these mediate calcium-dependent cell adhesion. They often hold similar cells together, examples of which are epithelial cells. Integrins have two membrane-spanning chains called alpha and beta. They bind and communicate with the extracellular matrix. They also play a very important role in cellular signaling and can greatly impact cellular function by promoting cell division, apoptosis, or other processes. Selectins are unique because they bind to carb molecules that project from other cell surfaces. They're the weakest formed, and they're expressed on white blood cells and endothelial cells that line the blood vessels. We had a lecture entirely on the immune system in our biology unit, but it's important that we mention antibodies and immunoglobulins again, because these are also proteins, and they have obviously very important cellular functions. They target specific antigens, and they retain this information for later use. Immunoglobulins and antibodies have a very similar form. They both comprise of a variable and a constant region, and most of them are held together by tertiary bonding structures. Now that we've gone over the cellular functions, we'll go into biosignaling. The first type of proteins used in biosignaling are what are called ion channels, and there's three kinds. There's ungated, voltage-gated, and ligand-gated. Ungated proteins form channels, and they're constantly open, so specific substrates are allowed to go to and from these proteins. There's no mechanism that keeps them open or closed. Ligand-gated and voltage-gated have different mechanisms, so they'll actually remain closed until either a membrane potential hits it with respect to voltage-gated or a ligand binds to it. When this happens, it'll open up and allow for substrate to move through. Note that these are also substrate specific, so there's only a very select amount of substrate that can go through these. It's not just a completely open channel. Enzyme-linked receptors participate in cell signaling through extracellular ligand binding. This will create what's called a secondary messenger cascade. What this means is that when a cell gets the signal, when these proteins get the signal, it's not the signaler itself that's going to cause the internal mechanism. Instead, it's going to trigger the cell to execute its own mechanism for messaging throughout. The most common example that you'll see is cyclic AMP, or CAMP. G-protein coupled receptors are also enzyme-linked receptors, except they use G proteins and a GTP cascade to trigger this intercellular response. So we've gone over all of the biosignaling and functions, but how would we isolate these proteins in a lab? Well, let's go into some information regarding these processes. We'll start first with arguably one of the most used ways of protein isolation, which is called electrophoresis. This uses a gel membrane to observe protein migration in response to an electric field. What you do, as the picture suggests, is you would put proteins at one side of the gel. Once you run a signal or some sort of separation, the proteins will separate accordingly, leading to patterns like the one you see on the bottom right. There are different types. Native page maintains the protein shape, but because of this, the results are sort of hard to compare because this mass-charge ratio differs for each protein. If you want to preserve your sample for future use, you would have to use the native page. However, if you wanted a more accurate measurement, you would use the SDS page. But if you do use the SDS page, you're going to ruin your sample because it denatures the proteins. This will also mask native charge. Because of these parameters, they're more accurate in terms of size comparison, but they ruin the proteins, so be careful before you use it. Isoelectric focusing uses pI as the separation mechanism. Proteins will migrate towards electrodes until it gets to the region in which the pH equals the pI of said protein, so obviously the gel is going to have varying pHs. We talked about chromatography in our general chemistry module, but let's revisit it because it can be used to separate proteins as well. It separates based on affinity to a stationary and a mobile phase, and there's different types. The one that you'll often see is the column chromatography, which is simply a column that separates based on running a mobile phase through a stationary phase of beads. Usually this is some sort of silica or alumina gel. The different types are listed below. Ion exchange will use charge. Size exclusion uses porous beads that have holes for a specifically sized substrate. Note that larger compounds will elute first because they don't stick to the pores. This is a huge distinction that you're going to have to keep track of. MCAT test makers often love to use this to confuse test takers. Affinity separates by specific binding to a given substrate. With respect to proteins, this is the most commonly used because you can use it to tailor specifically to a protein that you desire. Well, now that we've isolated these proteins, how do we analyze them? Let's go into that. Protein structure is often studied using X-ray crystallography, or XRD, and NMR. XRD shows 3D protein structures using obviously an X-ray beam. An image is listed at the bottom right. NMR uses a magnetic field to determine structure. Oftentimes, you won't see X-ray crystallography directly tested on the MCAT, but NMR will. So if you need a quick brush up on what NMR is and how to read NMR data, refer back to the organic chemistry spectroscopy module. Oftentimes, when you look at protein structure via NMR, it's going to be messy and not as clean as you would like. To the bottom left is a sugar molecule that has been run through NMR. It's not as easy to read, but you can still get a general idea based on the information. If we want to look at the direct amino acid composition, that is the primary structure of a protein, we can determine that simply by using a hydrolysis mechanism. The sequencing will obviously require some sort of degradation. So what that means is that you'll take the chain of amino acids in the protein and break them off piece by piece from one end to sequence it. The most common is the Edmond degradation, which is listed below. Take note that if you do an amino acid composition via this sort of sequential degradation, you are also ruining your sample. Measuring activity levels is often done by following a process of a known reaction. So at the bottom there is an MHL reaction listed. So what this is measuring is how much of a product versus a reactant is present in given organs. By looking at this, we can determine how much of this protein or how active this protein is in creating a given product. If there's more reactant than product, we can safely say that it's not very active in that area, and vice versa is true if there's more product. The final type of analysis that we have to look at is protein concentration. This is often done by UV spectroscopy or color change reactions. Color change reactions can include BCA assays, Lowry reagent assays, and Bradford assays, but what this simply does is it changes color based on given concentrations. BCA assays and a BCA RAC protein assay, which is a variation on the BCA assay, are listed below. When we look at these color changes, we can basically look at how these proteins interact with this solution, and that interaction is going to create specific colors. So obviously, based on the color, we can determine the given concentration. We'll finish our discussion there. In the next lecture, we're going to start looking into some specific types of molecules and how they function.